This is our stop motion project of muscle excitation and contraction by Nicole Henkel and Ava Hill.
The skeletal muscle is a muscle attached to the skeleton bones that allow the limbs to move. The tendon allows the contraction of the muscle to make the final tangible movement. The paramecium is a sheath of connective tissue that groups muscle fibers into bundles. Fascicles are a group of muscle fibers bundled into one unit within a muscle that determine the kind of movement it can make. The epimecium is a sheath of connective tissue that surrounds an entire muscle. The endomecium is a layer of connective tissue that ensheaths each individual muscle fiber and separates single muscle fibers from one another. The sarcolemma is a specialized cell membrane which surrounds striated muscle fiber cells. Myofibrils are long filaments that run parallel to each other and combine into muscle fibers to make muscles. Muscle fibers are fibers consisting of a single muscle cell that in a group control organized movement. The neuromuscular junction is a synaptic connection between the terminal end of a motor nerve and a muscle. Synaptic vesicles are small membrane-bound vesicles at a synaptic junction of neurons that contain a neurotransmitter. The synapse is the site of transmission of electric nerve impulses between two neurons. Acetylcholine receptors are membrane proteins that bind the neurotransmitter acetylcholine. Calcium ion receptors bind the actin filament to expose the binding site for myosin heads to bind in order to stimulate muscle contraction. Actin is the thin cellular protein found in a myofibril of a striated muscle that works with myosin to form muscle contractions. Myosin is the thick cellular protein found in a myofibril of a striated muscle that works with actin to form muscle contractions. The H zone is the center of a sarcomere band that is only the thick or myosin filaments that become smaller as the muscle contracts. The Z discs are the side boundaries of a muscle sarcomere and also the attachment sites for the thin filaments. The neuron is a nerve cell that conveys impulses from the nervous system to a muscle. The axon is the long process of the motor neuron that carries electrical signals away. The sarcoplasmic reticulum is a specialized smooth endoplasmic reticulum structure in muscle cells that stores calcium ions. Excitation coupling. First, an electrical signal travels through the nerve to the neuromuscular junction. Next, the motor neuron will release the acetylcholine into the synaptic cleft. After that, the acetylcholine will bind to the receptors on the muscle fiber. Once bonded, the action potential then is triggered across the sarcolemma. The action potential will continue to travel across the sarcolemma through the T-tubules. Then, the T-tubules will activate the release of the calcium ions from the sarcoplasmic reticulum to the sarcomere and muscle contraction can begin. Muscle contraction with sliding filament. First, the sarcoplasmic reticulum releases the calcium ions into the sarcoplasm. Then the calcium ions bind to the proteins, the troponin and myosin, on the actin filaments. The actin will then change from a blocked to an unblocked position, and myosin heads attach to the newly exposed binding sites on the actin. After that, myosin will then pull the actin filaments toward the center of the sarcomere. ATP provides the energy for the myosin heads to simultaneously release and bind to the actin. Muscle contraction will then occur. To return back to the resting state, the calcium ions will be reabsorbed into the sarcoplasmic reticulum. This then causes the myosin heads to be blocked from the actin, and myosin's hold is released onto the actin, making the muscle cell relax and lengthen.